Okay, right now it is 5:33 p.m. on March 19th, 2020, and today I am going to do another rap video. Last time I did chemistry, I also did physics, and now I'm gonna do biology rap. And this time it's going to be the biology electives, meaning that we have the core part. We have like 24, 25, 26, 30 something, 40 something topics in the core part, and then we have the electives where we basically have to choose 2 out of 4 elective topics, and we sort of study that, and in the exam in paper 2 we are going to give a stack of papers, and they're actually 4 stacks, more like 4 questions, and we need to choose 2 out of the 4 questions. And for the 4, we have number 1, human physiology, regulation, and control, also known as homeostasis. For number 2, we have applied ecology, which we have also studied. And then for number 3, it's microorganisms, which is rumored to be the toughest one. That's why our teachers never taught us that, because our teachers know that it's too tough. And then we have book 4, which is biotechnology. And this one is necessary. Everybody had to choose this one because this is really basic. But between 2 and 3, 2 is actually tougher, I think. But if you use your common sense, 2 is also easier to understand in a way. 3 is way weirder and also less interesting. But also 2 is also not very interesting, kind of boring, and you have to write a lot for 2. So anyway, I picked biotechnology. But we studied both ecology and biotechnology and see which one better suits us. Well, biotechnology is more of my thing. But anyways, we're going to start off with homeostasis. And there are actually 4 chapters in homeostasis itself. And there we go, 4 separate chapters inside the book. We have the regulation of water content, aka the ADH chapter. We have the body temperature chapter, we have the gas content, and we have the reproductive cycle chapter, which is easily the best. Okay, this one is the ultimate best, and you'll know why. Because the 4th one is the easiest. I mean, I'm not a girl, okay, but this is easily the most simple. Anyways, let's start off with the first one, water content regulation. So, first of all, we can't drink too much water because too much water means water intoxication. But that's besides the point. We have the urinary system, which is basically, here are some blood vessels, but the blood vessels, they don't connect to our pee-pees. They actually go into the kidney, and then from the kidney to the urinary bladder is the ureter, and then from the urinary bladder to the outside world is the urethra. And then we have kidneys, which is the main point of this video. This is a kidney, and this is a diagram of a kidney. Now, there are a lot of nephrons in the kidney, and both blood vessels, arteries, and veins, they both go in and out of the kidney. But actually, if you know what the hell are arteries and veins, you know that the blood content goes from the arteries into the nephrons and then out in the veins. So there's that. And in the nephrons, we go even more micro-sized. There are these things. Now, first of all, there are several parts to this. We have the afferent the afferent arteriole, which then goes into a glomerulus, a highly concentrated bunch of just blood vessels with high pressure, and the blood content are sort of forced out of this thing and they go into the kidney tubules, the proximal kidney tubule, and down the loop of Henle, and then up again to the distal convoluted kidney tubules, and then into the collecting duct. And then we also have a bunch of arteries, a bunch of capillaries sort of surrounding the loop of Henle and the kidney tubules. And the reason being is that while all the blood content sort of forced itself out of the blood vessels into the Bowman's capsule. See, now you know how hard is biology. Actually, a lot of those substances get reabsorbed. So now we zoom even closer. We have the glomerulus and the capsule. As you can see, glucose comes out, amino acid comes out, water, salt, urea, everything sort of comes out. And then later on, they all sort of get reabsorbed back. The two conditions is a high pressure and permeable wall of the Bowman's capsule and also the blood vessel. I guess permeable would be the right word, even though, you know, if red blood cells somehow make it into the Bowman's capsule, you're fucked. And then we have the friggin' kidney tubules, just like in the intestines, just like friggin' air sacs, very highly folded, and yeah, just like brain neurons, intestinal villi, alveoli, capillary network, kidney tubules, umbilical cords, highly folded, lots of villi, increased surface area, steepens the concentration gradient, makes it efficient and continuous. Basically the material, they get reabsorbed back to the blood vessels, but not all of them. Roughly half of the urea goes back, but like half stays. Okay, yeah, half the urea, all glucose goes back. If there is glucose in your pee, you're screwed. Also, all amino acids go back. Also, most salts go back, and most water go back. Some water needs to stay to produce urine. You can't just, you know, it must be, you know, there had to be some sort of water. Now, they go back, and glucose goes back by diffusion and active transport, and water goes back by osmosis because of water potential. And we are already 8 minutes in. Goddamn. All right, Osmo Regulation. Now we are at the interesting stuff. We have the ADH, the antidiuretic hormone produced in the pituitary gland in our brain. The posterior, the anterior, posterior. That's the most of the hormones comes out of the posterior. So there's that. So say the water potential of your blood is lower than normal, meaning that it is thicker, it has less water and more, you know, nasty stuff. Then what it's going to do is it's going to release more ADH to make the wall of the collecting duct more permeable to water, and a greater proportion of water is reabsorbed in order to make the blood content less, or blood water content less, blood content less concentrated, more diluted, and that is very useful. If your water content is too much in your blood, too diluted, then you're gonna pee more of them out of your body. Okay, we have the dialysis machine, as you can see, which can do stuff. We have the dialysis tubing, which is differentially permeable. We have the dialysing fluid, which has a similar water potential. Otherwise, you know, you're gonna die. Same concentration, well, similar concentrations of nutrients, and has no metabolic waste. Okay, that's chapter 1. you know. Now we move over. See, now you know how tough it's biology. Now we move over to Chapter 2, Regulation of Body Temperature. This one is slightly simpler, I have to admit, but also not at the same time, because there's the too hot and then there's the too cold. And you can lose heat in your body by radiation, conduction, evaporation, and convection. Sweat by evaporation. And then you have the peripheral body temperature and then the core body temperature. Say your fingers can feel cold, but it doesn't mean your chest or your liver feels cold. You understand? There's the heat loss center in the brain, and then there's the heat gain center in the brain. In the hypothalamus. And okay, so three things that will happen. If it's too hot, then okay, let me show you the structure of your skin first. We've got a few things: the pressure receptor and pain receptors. We got the subcutaneous fats, triglycerides. We got 3 layers of skins here. Hair. We got the muscle pulling the hair, which is called the erector muscle. We got the sweat gland, and we also have the sebaceous gland, which is also connected with the hair. And we got the dermis, which is basically the little layer under your skin. Then we have the epidermis. Okay, when it's cold, your erector muscles contract to pull the hair upright, aka give yourself goosebumps, because they trap a thicker layer of still air. If it's hot, then the muscles relax and the hair goes back down. Number 2, if it's cold, sweating reduces. If it's hot, you sweat more. If it's cold, vasoconstriction of arterioles, where more blood goes into the shunt vessels and less blood goes into the skin blood vessels. And yeah. And then if it's too hot, then vasodilation. That's why your skin is more red under hot conditions. And we also shiver, which is called thermogenesis. If it's too cold, we shiver. Our body basically forcing us to generate heat. Now we have a hormone called the thyroxine in here, right below the larynx. We have the thyroid gland. So from the hypothalamus, it stimulates the pituitary gland, the posterior pituitary gland, to come out with thyroxine stimulating or thyroid stimulating hormone, and then the thyroid gland produces, secretes thyroxine. And what it does is that it increases the metabolic rate of body cells, so more heat is generated to keep the body warm. Yes. That's all you need to know. Oh, is there another thing? I bet there's another. There we go. Heat stroke and hypothermia. I haven't had a heat stroke before, but I know someone who does. And so I don't know how it feels. I really don't. But apparently if it's too high, then the mechanisms of body temperature regulation breaks down, and you sort of just passed out because you pass out. Yeah. All right. Now we move over to our 3rd chapter. Regulating gas content. Now this chapter is fucking annoying. Damn. Have I mentioned this? If you have a baby inside you, you have 5 circulatory systems in your body. Damn. But normally people have 2. What's special about regulating gas content is that it's not really a matter of hormones, because the center of your brain that controls gas content is actually in the medulla oblongata, which isn't which isn't really, in the cerebrum, cerebellum, or the pituitary gland. Okay, the basic rhythm of breathing, really, really simple. Intercostal muscles contract, diaphragm also contract, and your lungs, your thoracic cavity bigger, your lungs bigger because the air pressure in your lungs is lower than the outside. The outside air sort of rushes in, and then opposite when you sort of exhale. And rate of breathing equals the volume of air per minute, also equals the number of breaths per minute times the volume of air per number of breaths. CO2 concentration in blood. If there are too much carbon dioxide in your blood, which is an acidic gas, and it can react with anything H plus related in your blood to form carbonic acid, there will be, I'm already tired. There are chemoreceptors in aortic and carotid bodies that detects the pH level of your blood, and it sort of sends signals to your respiratory center. And so slower and weaker contraction of intercostal muscles, and then the rate of depth of breathing will decrease. Yeah, so if you breathe in too much carbon dioxide, your body is going to force you to not breathe too much. It's going to try to suffocate you, man. The heart. Right here you got the SA node, aka the pacemaker. It sends a signal to the AV node, which sends a signal to the muscle down here, and it sort of contracts. And it sort of pumps them out. God. So this 1 to 3 to 4 is basically the ratio of the first pump, the second pump, the first pump, the second pump, and then the rest, which you can also see in here. This is basically blood pressure in different chambers of your heart: right atrium, right ventricle, left atrium, left ventricle. Your right ventricle goes up to your lungs, your left ventricle goes to the rest of your body. That's why the muscle around the left ventricle is thicker, to give it more force to pump it all over your body. Goddamn, it's confusing. We have the nervous control of the output, if you can see, and we also have the hormonal control. That's smooth. The hormonal control of your cardiac output, and you can also mix them both. It can be both, But basically, if you're super anxious, scared, and you need to, say, get super excited real quick, what's going to happen is that your brain, your medulla oblongata, will immediately signal your pacemaker. No hormones included. But if the process needs to be more gradual, say I'm about to take the fucking DSEs, then the medulla oblongata will basically signal the adrenal gland above my kidneys, and then it will secrete more adrenaline to my blood, and then my pacemakers just get signaled. That's basically it. Or my pituitary gland secrete adrenocorticotropic hormone to signal my adrenal gland to secrete more adrenaline. That is also possible. If you want to increase your heart rate, you can do exercises, which I definitely lack, and basically there's that. Apparently, if you are an athlete and you're doing exercises, your cardiac output is 20,800 milliliters per minute. But if you're a non-exerciser, say me, you have 100 more cardiac output, which seems like a good thing, but actually it's not good because your lungs, they need to pump way faster and your diaphragm gets cramps and you can't breathe. And that's basically what happens to me every time I run. My diaphragm just goes insane and I just collapse to the ground and die. Okay, finally chapter 4. Cuatro. Hell yeah. Reproductive cycle, aka the menstruation cycle, the very iconic, famous menstruation cycle. Now, this is going to be complicated, but also incredibly fun. 4 things here. First off, your FSH and LH levels, your follicle stimulating hormone and your luteinizing hormone. They're there the whole time. So it's sort of, because we have the follicle stimulating hormone, your follicles start to develop and start to mature, and because follicles start to develop, the follicle secretes more estrogen, so the estrogen levels rise. But because estrogen actually inhibits the level of FSH and LH, they sort of okay, wait. First, the estrogen makes your lining, your uterus lining thickens, right? And progesterone maintains the thickness. And then afterwards, because it hits a very sharp peak, it also somehow stimulated both peaks of FSH and LH, even though that isn't supposed to happen. But I guess it just passes a threshold of sorts. And because of a peak in LH, the luteinizing hormone, ovulation is triggered. So on day 14, the follicle breaks apart into a corrupted follicle and the ovum, and it becomes a yellow body. And the yellow body secretes more estrogen and more progesterone. And then because the level of estrogen is higher again, it inhibits FSH and LH, so it remains low. And then the yellow body sort of degenerates, so the level of both estrogen and progesterone gets lower, the lining becomes thinner, and the whole cycle starts over again, and this thing goes up again slightly. Damn, okay. I'm already getting all hot right now because I'm using so much of my mind power right now. If you're pregnant, successful fertilization, then your uterus lining will continue. Your uterine lining. Fuck. Your uterus is not uterine, right? Your uterine lining remains thick, and there will be a hormone called the human chorionic gonadotrophin hormone, the HCG, which works like the luteinizing hormone, and it prevents the degeneration of yellow body. And that's basically it. And then afterwards we have a bunch of different methods to contraceptive hormones, to contracept. You can take pills, synthetic progesterone, or synthetic progesterone plus synthetic estrogen in order to inhibit FSH and LH levels. And there are also many different reasons why you're infertile. Say if you're a dude, you don't have enough sperms, your sperm motility is poor, meaning that it can't swim up to the ovum in the oviduct, and also your sperm has structural defects. If you're a woman, maybe it's because your ova failed to manage or be released from the ovaries. Maybe it's because your oviducts are blocked. And maybe there are conditions in the uterus that didn't allow the implantation of the embryo, which means, hey, maybe they fertilized, but it never really implanted in the uterine lining, so which really sucks. But that's basically it. Now we move over to E4 Biotechnology, where there are actually only 3 chapters here, not 4. Thank God. And these chapters are Techniques in Modern Biotechnology, Applications in Biotechnology, and Bioethics, which is incredibly boring but somewhat important. So, biotechnology, we got the classical one. You brew wine, make cheese, bake bread. And then there's the modern one. Genetic modification, cloning, yada yada. First off, we have the recombinant DNA technology. I'm pretty sure everybody has studied this part. Basically, you find the human cell, you get the donor cell, you extract the DNA, and then you get the bacterium where we have DNA strands, and also the plasmids, and then you cut these two with the same restriction enzyme, and then you connect them again with DNA ligase, and then you insert it back into the bacteria, and then you insert it back to the bacteria by either heat shock with over 95 degrees Celsius or an electric shock, and then afterwards you don't know if it's successful or not, so you do the replica plating, where you put the antibiotic resistance gene into the plasmid and see if that antibiotic resistance gene somehow bursts between the desired part of genetics in your plasmid. If it does, then, or more like, okay, that's a special case. That's a small chance. You insert an antibiotic resistant gene into your plasmid, and then you put it on an agar plate with antibiotics, and if they still somehow grow, then hey, it's successful. If they don't, it's not successful. That's how it works, man. There are different advantages. Insulin produced by GM pure, the product yield is much higher, and the cost is much lower because bacteria, they multiply, they divide, binary fission, they do that all the time themselves. So we have a bunch of enzymes. RNA polymerase basically makes an mRNA. DNA polymerase basically connects 2 strands of RNA to become 1 strand of DNA. We got the reverse transcriptase, which reverse transcripts. We have the DNA helicase, which makes 2 strands of RNA twist. Oh, so DNA polymerase actually extends DNA, I guess. And then we have the DNA helicase. We have the DNA ligase, which connects, and we have the restriction enzyme. There are hazards, of course. There are benefits and there are hazards. When it comes to GMing things, like GMOs can outcompete the wild types. They may transfer their genes to the wild. Antibiotic resistance can happen. And then we have plant and animal cloning. For plants, you basically extract a very sterile plant tissue, and a bit of callus will form, which is basically a bunch of undifferentiated cells, and then you separate them, and then they'll grow the same things. And for animals, slightly different, similar concept though. You find a donor sheep, and you get a mammary gland cell, which is a highly differentiated, or any highly differentiated cell, right? And then you take its DNA, genetic information, and put it in a surrogate mother's ovum. They form embryo, boom, born. And this is identical. Well, this and this, the offspring. Except this is as old as this when this is born. When it's born. Yeah. Because of the telomere attached to all of its chromosomes in all of its cells in its body, which sort of counts time. It's like a ticking time bomb in your body. We got the PCR, the polymerase chain reaction. Hell yeah. First of all, we got DNA denaturation. Okay, I can spit, and you can take a little bit of DNA from my saliva and sort of copy it over and over again and expand it by doing this. So first of all, you denature the DNA, turn it into 2 strands. So you need to break the hydrogen bonds. That's why you really need high heat. Then you anneal the primers, lower heat, but still kind of hot though, 50 to 65 degrees Celsius. And then DNA synthesis, where there are a bunch of free nucleotides, and these primers, they stay there, but the nucleotides, they sort of attach and they go and they create new strands of DNA, which is really cool. And that is how you copy and paste a bunch of DNAs, apparently. And then we got the DNA fingerprinting. There are some variable number tandem repeats. And then you cut these repeats with restriction enzymes, and then you put them in some samples, and then you put them in an gel electrophoresis, and you sort of start it, and there's a nylon membrane. You started it already. They sort of move to the cathode because the larger the DNA fragment, the slower is it going to travel to the opposite side, to the plus side, because DNA has phosphate ions which are negatively charged, so they travel to the positive side. Basically, that's end of story, and you can see similar patterns by matching positions and stuff. And yeah, you can also plant some DNA probes, some radioactive DNA probes, to make sure that there are some VNTRs. There is a PCR-STR, polymerase chain reaction, short tandem repeats, which is the better version of RFLP, which is very annoying. RFLP, yeah. I forgot what does that mean. Chapter 2. God damn. You can create human insulin, growth hormones, vaccines, and monoclonal antibodies. There are different types of gene therapies. There's the somatic gene therapy, and then there's the germline gene therapy, where you do gene therapy on your pee-pee, and so those genetics are inevitable, which actually sucks, because it's like, oh, I'm deciding all of my sons and grandsons to have this character, which they normally wouldn't have, but I want them to, so I guess you can do that. We have the in vivo, we have the ex vivo. For the in vivo, you insert vectors with the normal genes into your body and sort of try to infect your genes, but there are problems with that. Or you can do ex vivo, you extract cells from your body and then have the vectors, aka virus, sort of infect your cells and then put those cells back in. Why the hell not? You can also do the stem cell therapy, where basically you do that to a stem cell. Yeah, you can do that to a bunch of stuff. You can do transgenic plants and animals, and yeah, I need to be fast. God damn it. Okay, I'm just gonna skip a bunch. Bioethics, toxic, biohazard tomatoes right there. Okay, so bioethics. Say GM. There are safety issues, ethical issues, like you can't play the role of God. Maybe you're trying to avoid eating for some religious reasons, like if you're a Muslim, maybe you don't want to eat pork. And then there are some environmental issues as well. For cloning, it's even more problematic. There are some ethical issues, economic issues, environmental issues, and legal issues, because nobody knows what the hell is going on. We have the Human Genome Project, which is sort of almost like a breach of human private information, because it's like, oh, so you have this serial killer gene, so you're a serial killer now. What? We have the gene therapy, and there are a bunch of ethical issues, legal issues, social issues. Like, oh my gosh, like if rich people can do gene therapy, rich people are too cool now. What? And that's basically it. God damn it. We still have like a bunch of other stuff. All of these is basic. Well, I have done genetic gel electrophoresis, DNA fingerprinting. I've done that in a practical, in a biological laboratory before. So, I have a stack of papers just highlighting and talking about how to do that, but that's basically it. Okay, I need to go. Bye.